Our first standard for the muscular system will cover the sliding filament model of muscle contraction. This is going to discuss what are the mechanisms for squeezing a muscle, for condensing a muscle, and then what are the mechanisms for relaxing the muscle. So we're going to talk about that coupling of relaxation and contraction. The standard reads, I can explain the sliding filament model of muscle contraction and covers section 9.1 and 9.2 in your textbook. Okay, a brand new heading for today, Unit 6, the muscular system. Muscle tissue is divided into three primary categories. First, we have skeletal muscle, which is the one that moves your bones. It's attached to the ends of those epiphyses and allows for locomotion. These are voluntarily controlled, meaning your brain sends messages through your nerves, through your neurotransmitters, telling these muscles to contract and telling them when to relax. Cardiac muscle makes up your heart. So this is responsible for the heartbeat that happens every second of your life. So this muscle is constantly relaxing and contracting, and it is involuntary. So even though you can do things that speed up your heart rate or slow down your heart rate, ultimately the pumping of blood throughout your body is going to happen without your ability to control how fast that's taking place. Finally, smooth muscle exists in the walls of your internal organs, like your intestines, and it gives you the muscle power that you need to push food particles and to push waste through your body system because it's not just going straight down or straight through you. We can't rely on gravity. We need muscle power. And also, these are things you don't control. You don't decide how quickly that food moves through you. It is an involuntary movement. We're going to focus on voluntary movements today, so we're going to spend most of our time discussing skeletal muscle. So muscle.1, first we need to highlight the structure of a skeletal muscle. Skeletal muscles have really long cells. So this tissue is not made up of a whole bunch of tiny blocks like maybe our epithelial cells, made up epithelial tissues, or some of our connective tissues. Instead, these are going to be super long cells that have multiple nuclei. Each cell is covered in a protective sheath called a sarcolemma, and then they're all kind of bundled together, each containing sarcoplasm, each containing their own set of organelles, and they're put together into these bundles. There are two primary muscle fibers that we need to learn the names of. Actin is a thin fiber, and myosin is a thicker fiber, and those are going to be the two primary tools that we use in order to contract a muscle. We call one of those bundles, one of those structural units, a sarcomere. And we'll highlight the anatomy of a sarcomere on the next slide. But when you look at muscle, you'll see a very obvious striped pattern as those sarcomere units repeat. And what you're seeing is thin filaments in the light sections and thick filaments, or myosin, in the dark segments. And as these sarcomeres repeat themselves, you can see there's a very clear repetition. There's a pattern going on. You'll see that striping. Okay, in your notes, I would definitely draw picture B here. Both the banding colors, where they're light and dark, and then all of the labels as well. So this is anatomy of a sarcomere. We said on the last slide that actin is a thin filament, meaning a very thin fiber. Myosin is a thicker filament. And so on this picture down here below, the thin filaments are represented in blue, and the myosin thick filaments are represented in this darker purple color. Now, you'll notice that there are regions where these filaments overlap, and there's regions where it's only myosin or regions where it's only actin. We also have these crazy Z lines down either side as well. So there are names to all of these bands that we can see. So the I band is your lightest band, and it is only comprised of actin. So that is in between all of the myosins. Right down the middle is a Z line, but in between those myosins we have I bands. A bands would be anywhere we have myosin. So this entire expanse would be an A band, and then we repeat. We have another A band starting here on the right, and another A band starting over here on the left. Through the I bands, we have those Z lines. They're sort of like anchors for the actin as it extends out towards the myosin. but you'll see these very clear zigzag structures that mark the ends of a sarcomere unit. So from Z line to Z line, that is one sarcomere. The H zone is the area in between the actin filaments where we only have myosin. So where there's this gap, that is known as the H zone. And it's going to be really important when we're talking about muscle contraction to note how big the H zone is or whether it's even there at all. Finally, the M line is a structure not labeled on this picture, but it goes down the very center and it anchors all these myosins together. So this line right here would be your M line. Okay, so we have our thick filaments composed of myosin, and these myosin, if you look even closer under a microscopic image, they're not just smooth filaments. They have these little heads on them, little arms almost, which are capable of reaching out and grabbing actin. And that's what they want to do. However, you'll notice in this picture, they're not touching actin. The thin filaments, of course being our actin, are represented here in gray. So these little bead-like structures, those are our long actin molecules. The reason that the myosin and actin are not connected is because actin is protected by two other filaments, a combined molecule called troponin and tropomyosin. Troponin is this small blue bead-like structure. Tropomyosin is the very long strand. And together, these things cover up the binding sites on actin so that your muscles are relaxed most of the time. Because if these myosin heads were allowed to grab the actin, you would have a contracted muscle, but you would never be able to relax it. And most of the time, the majority of your skeletal muscles are not contracted. They are relaxed. It takes effort to contract them. So that troponin and tropomyosin complex is very important. Okay, here are the steps for how we contract a muscle. And you'll notice we're starting in the nervous system. So let's say you want to move your biceps. You want to lift something up. That message is going to start in your brain. You're going to consciously make that decision, and you're going to send that decision down the length of your nerves to the very end where the nerves meet the muscles, and that is called a neuromuscular junction. When muscles get stimulated by a motor neuron, that's when they know to contract. So without the nerves telling them to do so, these skeletal muscles are going to stay relaxed. So what's the sequence of events? Well, first, the nerve impulse releases what's called acetylcholine. It's a neurotransmitter that's abbreviated ACH. So in this upper picture, we see these little vesicles containing dots. Those dots are the acetylcholine neurotransmitters, and most of the time they're stuck in here. However, when this neuron gets a message from the brain that says, "Ooh, time to contract," then those vesicles will get released out into the space, and ACH will flood the space between the nerve, which is this bulb, and the muscle, which is represented all around it. ACH will float for a little bit, but it will very quickly find receptors on that muscle cell. So there are receptors all along this surface that are waiting to bind to ACH so that they can start the process of a muscle contraction. When that happens, it causes a change in the permeability of the membrane that allows different salts like sodium and potassium to generate a muscle impulse. We'll talk a lot more about sodium and potassium when we discuss the nervous system, but for now, just note that these two salts are involved in starting a muscle impulse. Once they do that, there is calcium that will get released out into the sarcomere region, and that is what will really start our muscle contraction. So sodium and potassium will start to flood parts of the muscle, but they will cause the release of calcium, which really gets the mechanism going. Okay, step 2. What's going on inside the muscle? It's called excitation-contraction coupling. So this is how that stimulation leads to a muscle contraction in which the muscle has gotten shorter. So when it's relaxed, calcium is tucked away in a region called the sarcoplasmic reticulum. It's like the endoplasmic reticulum of the muscle. Also when relaxed, that TT troponin tropomyosin complex is covering the binding sites of actin, so there is no binding between the thick and thin filaments. When we stimulate the muscle, however, we do the opposite. That stimulation causes the sarcoplasmic reticulum to push out calcium molecules. Calcium is more attracted to troponin than troponin is attracted to actin. And what I'm saying there is once that calcium gets pushed out, once it gets released, it's going to find that little molecule of troponin, and when it binds to it, it's going to cause tropomyosin to move. That binding causes a little molecular rearrangement that forces the TT complex off of actin. Now that TT has bonded to calcium and has moved, that leaves its binding sites open and ready for myosin to bind. Myosin is going to do just that. It's going to very quickly notice that those binding sites are exposed. It's going to connect to those binding sites. It's going to attach to them. And it will start to, based on just the geometry of this muscle, will start to pull backward and will shorten the length of the sarcomere. And as this happens down your entire muscle, you have this shortening and this pulling that will contract the entire thing, will make it shorter, all because these tiny myosin heads have attached and pulled on the actin filaments. Step 3 is called cross-bridge cycling. When the myosin binds to actin, it releases two molecules that it has been storing up, ADP and another phosphate molecule. In its place, a new ATP molecule, which is incredibly energy rich, binds to myosin. And it's really important that we bind this ATP molecule because it will provide the energy for us to relax the muscle, to break that cross bridge. If we didn't have this ATP, we would go through essentially what rigor mortis is, when your muscles contract and stiffen up and they can't relax. That's because they do not have the energy to do so, to rip these myosins off of the actin to which they're so attracted. Once that energy is used up, the ATP breaks down into ADP and phosphate, and you'll notice everything looks roughly the same except we got to get that calcium off of there. When we relax the muscle, we'll pull the calcium off and everything will return to its original state. TT will cover actin and the myosin will have ADP and phosphate. So, relaxation. How does this happen? Well, we have to cut off the message at the source. So we said acetylcholine started and triggered this entire reaction. So we're going to send an enzyme in called acetylcholine esterase to decompose the ACH and stop the impulse. Without the impulse, calcium will be forced back into the sarcoplasmic reticulum and the TT complex can resume its place covering those actin sites. Your case study for this unit is about muscular contractions, and it is a condition called muscular dystrophy. We're going to talk about who is primarily affected, what are the symptoms, and how does this affect one's quality of life.